Hello my dear students, I am Dr. Vaishaly Bharambe, Professor Anatomy. Today I am going to teach you how to draw the microscopic structure of connective tissue. Are you that student who doesn't know how to draw well in histo journals? Or are you that person who doesn't even understand the corrections told by your teacher? Then follow this series to learn how to draw histodiagrams in your journals as well as in your exams. So today, microscopic structure of connective tissue. First, understand that connective tissue is made up of cells and extracellular matrix, which is made up of fibers and ground substance. You've got three items here: cells, fibers, ground substance. In your class, you must have been told there are many cells, there are many fibers, and so on. Just remember, variable mixing of cells, fibers, and ground substance results in various types of connective tissues. So, by variably mixing these three elements, you can draw loose areolar tissue, dense regular connective tissue, dense irregular connective tissue, and adipose tissue, which is what we are going to do today. Let's begin by drawing a section of the loose areolar tissue. This is the diagram we want to draw. As you can see, it contains a lot of cells, a lot of fibers, and a lot of ground substance. So I'll teach you the cells individually and then we'll put them together to draw the diagram of loose areolar tissue. Come, let's begin by taking our eosin and hematoxylin pencils and begin to draw the first cell, the fibroblast. So draw your fibroblast in such a way that it shows a lot of fine cellular cytoplasmic processes. The cell appears to be spindle shaped and the nucleus somewhat centrally placed but flattened. And that's how easy it is to draw your fibroblast. So this is what your fibroblast looks like. Label it well. Let's go on to now draw the next cell, which is the adipocyte. Draw the adipocyte as a somewhat roundish cell with a flattened nucleus at the edge, with minimal cytoplasm surrounding the nucleus. Observe how the cytoplasm forms something like a rim with a central collection of lipid in the cell in this manner. Okay, and that, my dear students, is your adipocyte. Label it well now with an eccentric nucleus, lipid droplet in the center, thin rim of cytoplasm. See how it looks like a signet ring. The ring that you wear in your hand with a stone, it looks somewhat like that. Let's now begin to draw the macrophage. Draw the cell membrane in this kind of such tiny folds. Each of these is a projection and this projection helps to engulf the foreign particle into the cell. You can see a central kidney-shaped or rounded nucleus. Okay, draw that well. Now draw the mitochondria. A lot of cellular lysosomes and vacuoles are to be drawn. The rough endoplasmic reticulum, the Golgi apparatus, and finally stain the cell. That is your macrophage. Now, my dear friends, this is your macrophage. Label it well. Label the pseudopodia, which are going to help to engulf foreign particles. Make sure you add a few foreign particles in the lysosomes which have been engulfed and are now in the forming a phagosome. Okay. Label the kidney-shaped nucleus, label the vacuoles. That, my dear students, is a macrophage. Let's move on to drawing now. The next cell we'll be drawing is a mast cell. Draw a somewhat rounded cell with a lot of tiny little vesicles, okay? Draw them dark and light, indicating that there are lots of such vesicles. Now in the center, draw a nice rounded nucleus, color it, stain the cell. That, my dear students, is a mast cell, okay? Remember that these numerous granules that are present inside the mast cell will release the cellular content outside, resulting in various reactions in the body. The next cell we want to draw is a plasma cell. See how large it is. Draw it well, large. At one edge of it, draw the eccentric nucleus. Stain it. Now let's draw the chromatin material. See how I'm adding the chromatin material in form of lines from the periphery so that the final appearance of the nucleus is like a cartwheel. What's a cartwheel? Bullock cartwheel. Okay, it looks something like that. Okay, now let's add the cellular contents. Add the Golgi apparatus. Add the rough endoplasmic reticulum. Why do you need so much rough endoplasmic reticulum? To produce the antibodies for immune response of the body. Okay, and there you have your plasma cell. Label your plasma cell well. Cartwheel nucleus eccentrically placed, rough endoplasmic reticulum, basically a large rounded cell. Students, let's now learn how to draw the collagen fibers. Remember, collagen fibers are eosin stained fibers which run in bundles. Individual fibers do not branch, however, bundles can branch. That's it. That's how simple it is to draw collagen fibers. Now, I'm not going to teach you how to draw reticular fibers. Just take a look at the images. Very fine fibers, they're also type of collagen fibers. These are reticular fibers. Elastic fibers are thin, fewer than collagen fibers. They run singly, they can branch and anastomose. That completes our requirement of some knowledge about cells and fibers. Let's now go on to draw the loose areolar tissue. This is the image we need to draw. See how it contains cells, fibers, and then the basic pink colored ground substance. Let's begin to draw this diagram of loose areolar tissue. So first you can see that I have stained the slide. A light pink ground substance is drawn. On top of that, begin to draw your collagen fibers. Okay, draw them in bundles going in various directions in this manner. Now let's add the elastic fibers. See how individual fibers are running singly and branching. Okay, there you have your fibers and you have your collagen and elastic fibers. Now the next thing we will do is draw the cells. So see how I'm drawing a spindle shaped cell. Okay, what does that mean? It means I'm drawing a fibroblast. You can see that it's closely attached to the collagen fiber which fibroblast produces. Okay, so fibroblast produces the collagen fibers. Draw a lot of fibroblasts, stain them, draw the nucleus. The next cell I want you to draw is a large plasma cell. Remember to draw your cartwheel nucleus and some cellular contents. Now let's draw a large lymphocyte here, a small lymphocyte here, a mast cell. Take your time. Draw the tiny granules well. Now let's draw a macrophage. Okay, remember this is a cell which has so many projections from it. Okay, draw those. Draw your somewhat kidney shaped nucleus. Draw the cellular contents. A few fibroblasts. And there you have your slide of loose areolar tissue. Label the slide well, making sure you have labeled all the cells, the collagen and elastic fibers, and finally the ground substance. This is a table that shows you the different types of connective tissue that we are drawing today and the comparative amount of cells, fibers, and ground substance present in them. So we just drew the loose connective tissue or the loose areolar tissue. You can see that there are plenty of cells, there are few fibers, and there is abundant ground substance. An example of loose connective tissue is loose areolar tissue. Let's now go on to draw the dense regular connective tissue. For dense regular connective tissue, you have to draw collagen fibers running in such bundles. Remember the rule, bundles can branch, but individual fibers must not branch. There are some spaces in between, as you can see. These spaces contain fibroblast cells. I'm drawing the nuclei. I'll tell you we'll be drawing the cells also later. Okay. So that is our dense regular connective tissue. In the gaps in between, we must add the blood vessels now. So add your blood vessels, the lining of the capillaries, the nuclei of the capillaries, stay in the cell, and there you have the slide of dense regular connective tissue. Label your diagram, making sure that you label the abundant collagen fibers running parallel to each other, the fibroblasts which are producing the collagen fibers, minimal ground substance, and blood vessels in between. Let's now begin to draw our third slide, dense irregular connective tissue. What does this consist of? It is also connective tissue, but here collagen fibers are in bundles in variable directions. See how you can see collagen bundles going in variable directions. In between these are the blood vessels, and of course between the collagen fibers there are the fibroblast cells. Now what is left? Take a look at the image and tell me what is left. Of course, the ground substance. Remember, if you do not stay in the slide, if you do not draw the ground substance, these are all things that are flying in the sky, which is not possible. So, finally, we stain it, and that, my dear students, is dense irregular connective tissue. Make sure you label your diagram: irregularly arranged collagen fibers, fibroblasts, blood vessels, and a minimal ground substance. Now I want you to take a look at this. This is dense regular and this is dense irregular. Both have few cells, mainly fibroblasts, but in dense regular there are lot of fibers which are arranged regularly. In dense irregular there are again lot of fibers, but arranged in an irregular manner. Okay, so that's the only difference between these two. They both have minimal amount of ground substance. Let's now draw the last tissue, which is the adipose tissue, which needs you to draw such cells, almost rounded cells, with cytoplasm collected at one end and nucleus clearly marked in it. Now remember, there are so many fat cells that you have to draw them one over the other. So now let's draw the next lot of fat cells, okay? Adipocytes, as they are so beautifully called. Draw again the cytoplasm at one edge. Draw the nucleus, flattened nucleus at one end. Fill up your fat cells with fat in the center, don't leave them empty, and draw a lot of capillaries in between them. Okay, these are capillaries. Draw the epithelium, draw the flat nuclei of the capillaries. Now take time to draw these elastic fibers, a few additional cells, and this is your diagram of adipose tissue. So this is your diagram of adipose tissue. See how these are large empty looking cells with a thin rim of cytoplasm at the edge. Fat droplets in the center. Nucleus is flattened and pushed to the periphery. Finally, in this table, you can see that the cells in the adipose tissue are adipocytes. There are few fibers. There is very little ground substance. So what is more? Cells. But they are predominantly adipocyte type of cells. I have also given you a little bit about mucoid connective tissue in case your medical school requires you to draw the mucoid connective tissue. That's it, students. I hope you enjoyed drawing connective tissue with me. I'll see you in my next video. Do like, share and subscribe to my channel and hit that bell icon in case you enjoyed this lecture. For more such videos, visit my website vbanatomy.com. Thank you.